Next up, we have our last speaker of the day. Kristen Nields is with us, and she is coming from J&J in the Innovation Sciences Group, and she will be giving us a little bit of a different talk, showing us the technology of the iCIEF and UV with the Intabio system that she has. So she's looking at she's going to give a discussion on reducing risk and accelerating cell line development using this technology. Kristen, I will hand this over to you so you can give us the update on your research.
Thank you. So good afternoon, good morning, good evening, everybody. I wanted to thank everybody for joining this presentation today as I walk you through how we've been implementing some new technology with the Intabio into cell line development and why it's become a primary tool for picking our single clone and how we can accelerate it for product quality characteristics. So today I'm going to give a background on why we use it, who we are, where we sit within the Johnson & Johnson Innovative Medicine Organization. I'll go through then the instrument functionality and then follow it up with some data analysis and how we are beginning to utilize this routinely in our workflow. So currently, we have a lot of challenges with identifying charge variants. So within our cell line selection team, we have to do a product quality characterization for all the clonal material coming through for all new modalities. So this is stuff coming from discovery that we want to take into development. And charge heterogeneity, as being a CQA, has always been quite a gray area on identifying what all of those charge variants are and how any process inputs are going to impact them downstream from us once we start to design a clinical process. So it's very important up front that we're trying to understand as much as possible what the charge variants are. So we have to be able to make an informed decision with CMC, when we go in front of leadership, that there will be no issues with safety, efficacy, stability, and we want to make sure that the method that we're putting forward can be robust and can set a specification for future QC release. So currently, we have the Intabio ZT system, right? So CIF we know is a specification requirement. We have many times a CIF will fail spec, and we have to drill down what that root cause is. It becomes a huge bottleneck. It can take hours upon days, upon months to try to get fractionated material from a CIF or a CEX system, and then to run it to peptide map to do a definitive ID on what that variant is. So sometimes we need to do that for each variant, set a specification. That can take a lot of time, a lot of resource. So it would be great if, we had a tool in place, and we've been talking about this in the biophysical space for years, where we could just, have a CIF-MS ID right away with a rapid turnaround, where we'd be able to make informed decisions rather than, creating the bottleneck in the system. So that's where the Intabio entered. So we started with the Intabio a few years back. And we started with just looking in the research space, and now we're taking it further into cell line selection. So it's a iCIEF-based platform, right? So we have the UV that's coming off the chip, and that is a correlation to our ICE or Maurice profiles. So we want to make sure that we have a complete correlation with our UV, and that everything aligns. So we're using the same pharmalates, the same PI markers. We have a direct mobilization of the separation from the UV right into the mass spec. And because we have it mobilized, we have pretty decent sensitivity where we can see down to 0.1. If it's like 0.1 mg per mil, we can usually catch a PTM in that range. It requires a small amount of material. and it only takes 30 minutes per sample to acquire the data. So it is, 15 minutes per injection, but then, the processing time and then the data analysis time on the back end, we're averaging maybe, not a super quick turnaround, but much quicker than we were before. But we're still setting up our workflows and our work streams, but we usually get data out within a week. So we can talk about how it functions. So we have the front end of the Intabio, and we put the chip on where we add our analyte, catholyte, immobilizer. We load the protein. It's separated by charge, imaged by UV, and then we direct it right into the 7600. So let's get into the nitty gritty of some of the data here. So what we've been doing for most of our programs right now is running Intact and dGly. And the reason we run the dGly is because we want to make sure we're getting an accurate quantification on the glycation. So glycation for us can be critical to some to some processes depending where the glycation is happening, but we do want to measure that level of glycation as well. So that's why we tend to run native and dGly for everything. We did see that sometimes the dGly will increase a basic peak, and that is also how we can pick up clips. So there's been a lot of information that we have gathered over the past 3 years on sample prep, different enzymatic digests, on how we can further streamline our characterization of these charge variants, which becomes pretty critical at time of cell line selection and also during a clinical manufacturing campaign, because we're able to go in definitively and without fractionation pretty much pull that apart and say, without a doubt, it could be deamidation, it could be glycation. So we have gone into a lot more work with using multiple sample digests and making sure that, when we call an unknown out, that we are pretty definitive and always trying to back it up with peptide map. But here in the Intact and the dGly, we see that the PI, shifts a little bit in the dGly, which we expect for deamidation events to take place. And then we have an acidic and a basic quant. So for this molecule in particular, there is a pretty nice size basic, and even with treated with carboxypeptidase, it could be a C-terminal lysine that we can, quant or remove it with carboxypeptidase and remove that basic peak. So when we compare the integrations between the Intabio and the Maurice, we're looking here how the Intabio is in the darker blue trace here, and then in the Maurice is in the lighter blue trace. So the reason we've tried to use both for our analyses is we get a better resolution between what we have noticed across several programs is we have better resolution on the Intabio chip than on the Maurice. And in turn with that resolution, then that makes the mass spec a lot more easier to identify. We deconvolute then after we get everything, and when we're in the native state, we'll get our typical sugar groups, our G0Fs, our G1F, G2Fs, and our A-gly species. However, when we do the because the G1F is exact the 162 Daltons, that's why we do the glycation, because we do want to make sure that anything coming out in that acidic region is glycation. Then going through in the heat maps, this is an example of how we drill down into the characterization. So going from top to bottom, basic to acidic, the blue being the basic area and the white in the main area with the red in the acidic region. We can see here that in the top area, we have some C-terminal lysine and PowerQ compared to the main peak that's coming out there. So then we can see that we start to have in this dGly sample a little bit of glycation popping up, and then you can see just by visually looking at the heat map that it predominantly turns into 100% glycation as we go into the acidic region along with deamidation. Our quantification, we take directly from BE. So we will take the peak intensities and compare and do a calculation with the peak intensity and the UV area, and do a global composition under every main peak, basic peak, acidic fraction. But then again, we will also take that UV total quant, and then under that UV total quant, we can tell if it's a deamidation or a glycation species and how much of that is contributing to each acidic main or basic slice. So the advantages of acquiring more information. So we are acquiring more CIF data, which helps us in the long run, especially with MS data to support that. We can advance on release testing, and where the bang for our buck comes in is really aids in investigative reporting. It can optimize the way we are processing things downstream from us, so it can help with process lockdown in clinical campaigns. It can aid in their development. We can definitely increase throughput by trimming back those investigations and actually looking to root cause analysis quicker, and getting batches out sooner. Back to our portfolio, another anomaly we saw during cell line selection, and a pretty interesting technique that we did pick up, was in the one molecule coming through. We had a basic peak that was kind of hidden under the main peak, and we did see a shift, and screening 24 clones going down to top 8 clones based on titer. We then look at those top 8 clones, and the one clone which had the highest titer had this basic peak. And the basic peak was a shift of 29 Daltons, but it was something we did not see in any of the other clones, and started to investigate, what that possible plus 29 could be, corroborate with our mass spec colleagues to see if they saw anything. And, it was stated that, we were still going to investigate it after cell line selection and after we picked the clone. And then a few months afterwards of us doing many other things to try to figure out what that plus 29 was in the basic area, we did see that it was an actual genetic mutation of a Q to R. So something that was a plus 29 shift and a basic shift, because you're moving that PI down to a PI of 10. It makes sense that it's in the basic region, and then the plus 29. However, the peptide map initially missed it because it is a very short peptide. So it eluded before even capturing it, and it eluded into waste before even capturing it into the UV space. So it was something where we said, okay, this is a really good tool that's picking out something. So how can we better use this now downstream going into all of our cell line selection? So what we do now is we run all of our top clones through the Intabio system, and if there is a unknown peak, we try to capture that data immediately, and corroborate with peptide mapping and additional studies if needed, if they're still not aligning. So it has shown its value on several programs so far, with genetic mutations, with color, with mispairing. So we have been able to put molecules on pause, put on hold, do further optimization because of the data that we're finding, and being able to give senior leadership an informed result. So in conclusion, we've been able to use the Intabio ZT as a powerful investigative tool with sensitivity to even low-level charge modifications. We've been able to quantitate O-length glycosylation in the hinge region of multi-specific molecules. We've been able to look at lysine hydroxylation in the FC backbone, disulfide mispairing caught prior to molecule entry into CLD, cyclization of N-terminal glutamate observed during accelerate stability studies, and advanced glycation end products looking at how we can dig into the color of our final drug product and what attributes are actually causing that color and how we can lower that to meet a clinical specification. With that, I will leave that for questions.